Hello, welcome to Converging Dialogues. This is Xavier Bonilla. On this episode, I'm excited to have Bill Sullivan on the podcast. Bill is the Showalter Professor of Pharmacology and Toxicology and Microbiology and Immunology at the Indiana University School of Medicine. He has a PhD in molecular and cell biology, and he studies many of the parasites that are responsible in our gut. He is the author of the book, Pleased to Meet Me, Genes, Germs, and the Curious Forces That Make Us Who We Are. And that is the book that we talk about on the episode. We start the conversation by talking about what is DNA and what are genes, and he gives a very nice, tangible way of understanding what those things are. We talk about the role of genes in environment and also epigenetics. We then get into some of the specifics. We talk about genetics of taste and caffeine. We talk about the genetic aspects of addiction in various substances. We talk about the genetics of appetite, then the gut microbiome. We talk about genes and emotional states, genes and beliefs. And we also end by talking about CRISPR and the ethical landscape of genetics. One of the wonderful things about Bill that is great in his book, it comes through in the conversation, is that he's able to make really, complicated types of terminology, types of study, really easy to understand without watering it down too much. So many people talk about genes now. There's many popular science books that come out and they talk about genes, but his book does a really nice job of, making it easy enough where a high school student or a college undergrad student could read this and get a really good understanding. He has lots of really wonderful stories and examples and makes it really kind of come to life. As I was reading it, I've talked to a bunch of folks and I've read a bunch of, papers and books on this, and it was still really, wonderful for me to read this. It was like, yeah, I know this, but I'm still learning a lot of things. So it's really got a nice, kind of wide range of people that know about it, people that don't know anything about genes and how they all work in the body. We have a really nice exchange in the conversation about epigenetics and some of his affinities to it and some of my critiques of it. And we have a nice conversation about that and really just talking about how genes are important to study, to know many things about humans and how we can look at how they interact with the environment and kind of the future of where this research is going. Bill is a wonderful guy. Really, really enjoyed talking with him and connecting with him. And so now I bring you Bill Sullivan. I'm here with Bill Sullivan. Bill, thanks so much for coming on the podcast. I've been looking forward to this and looking forward to talking about your book.
It's a pleasure to be here. I'm a fan of the podcast, so it's great to actually get to speak to you. Thank you.
Yeah, thanks. Appreciate that. The book is called Pleased to Meet Me, Genes, Germs, and the Curious Forces That Make Us Who We Are. Is this your first book or your second or third? Which popular book is this for you?
It is my first book, yeah.
Nice. Very nice. Well, it's very well written. I think genes kind of have this ebb and flow of interest for people in the public, and it's super, super, super readable. I could probably give this to, just any lay person. They could pretty much get it. So it's not big, bad, and scary kind of hard science stuff. There's other books out there that are doing that. And so I think this is super important because we need something like this to be able to talk to people in really, really, easy ways. So the book definitely does that. So it's great. It's really, really great.
Well, I'm really thrilled to hear you say that because that was my objective. I wanted to take these complex biological mechanisms and, inform people about some of the really fascinating science without boring them or confusing them. Mm-hmm. And if you just want to learn a little bit about yourself or how other people differ from you, that's what this book is all about. It's an attempt to utilize science in an accessible way so we can all understand one another and get along better at the end of the day.
Yeah, and you definitely, I think, accomplished that. Well, just tell folks, who you are, what you do, what's your day job when you're not writing books, and all of that wonderful stuff.
Sure. Well, the book is a logical extension of what I do. I'm a professor at the Indiana University School of Medicine.
Mm-hmm.
I've been there going on 20 years now. And primarily, I research parasites.
Mm-hmm.
So some of the parasites I do talk about in the book, because there's one in particular that we study called Toxoplasma gondii, which we might talk about,
Mm-hmm.
that infects about 1/3 of people on the planet.
Oh.
And according to some studies, might be affecting the way people behave. So that's a fascinating, parasite to me, and it's a thrill to get to go to work every day and try to solve these biological puzzles with some really outstanding trainees, students, and postdocs. And then the joy of getting to teach them as well as medical students is a thrill. In addition to that, so that's pretty much my day-to-day life. And the book kind of grew organically from some of the genetics background that I have, the microbiology background, and so on.
Yeah, that's wonderful. So you're doing mostly teaching and then writing, I guess, scientific papers, and you've written this, popular book. That's where the majority of it is. Is there other things in this world that you would want to do that you haven't been able to do? Like, maybe at a bigger level or something like that, or something that you were like, I'm too lost in like training and all of the like administrative stuff. I wish I could just do research, or, how do you kind of see it now of 20, 25 years have been doing it?
Yeah, that's an excellent question. And it's one that I constantly ask myself, because, as you get older, you always wonder what you want to do with the remaining time that you have, because we're all on the clock. Yep. And as you age, you become more and more reminded of that, as time goes by. But the bulk of my career has been research, and that's exactly what I wanted to do with my life. It's just a privilege to get to go to work every day and actually get paid for solving puzzles and mysteries. And, what person doesn't like a good mystery? Mm-hmm. So we work in the lab to develop, ideas about biological phenomena that we see. We try to brainstorm ways that we might be able to treat infections, and then go back in the lab and try to test those ideas. So over the course of 20 years, we've had some that didn't pan out, but quite a few that actually look very promising. And I hope with the time that I have remaining, that we can see some of those treatments to their logical end, see if they might actually work, in preclinical models or maybe even people, or identify novel routes that people can explore later on. But your question's good, because as my career has evolved, I've gotten more interested and passionate about science communication and outreach. There are a lot of scientists and physicians who don't really engage in those sort of activities. Mm-hmm. And mm-hmm. I find that to be a little tragic, given the environment that we currently live in. There seems to be, at least from my vantage point, an increase in the past decade or so in sort of an anti-intellectual, anti-science movement that is denigrating experts and expertise. And I think that's to our detriment. We really can't function as a democracy without trust in some of the institutions that make our country a free and wonderful place. So that to me is a little scary. And I want to be one of the voices out there who can communicate what we are learning about or what we observe in the lab, in the hospitals, in the clinics, so that people can not only understand what's going on, but we can engender more trust in experts and professionals, so that we can be properly informed when it comes to educating ourselves and making evidence-based policy so that our country can be competitive on the global stage.
Yeah, I fully agree with you. Unfortunately, there is a lot of science that's done that does not talk to many people well and doesn't communicate the findings. And when we don't do that, there's a void that needs to be filled. And what fills that void? A lot of the times it's a lot of pseudoscience, anti-science, conspiracy theories. And so, I think that part of the job people are realizing, with the, advent of the digital age and, technology and so much access to information all the time, that's a key part of the job too, is, how are people able to do that? And so you're doing it. And I think we're all doing it in the science field, hopefully more and more. So, it's great. So let's start with DNA. This is something that you explain early in the book and will be helpful for many of the other examples we use in the book, or that you talk about in the book. So how do you usually explain DNA, the A, T, C, and G, and how many genes are implicated in even the simplest things such as height or, even eye color? Most people understand now that, there's thousands of genes that are implicated in, different behaviors or for education, et cetera. But even very simple things, quote unquote simple, thousands of genes are implicated. So maybe just tell us what DNA is, how do you usually explain it, and how many genes are implicated for, even one or two traits.
Sure. There's a lot to unpack there. So if I forget something, please circle back and we'll try to address it in the end. But I think most people are aware that DNA basically builds an organism, including people, of course. So it's the recipe for life, if you will. And essentially what it does is DNA is comprised of genes. DNA is a molecule that is built into chromosomes as it complexes with proteins in the cell. And we have 26 chromosomes, half we get from mom. Well, no, scratch that. We have 46, 46 chromosomes, half of which we get from mom, half we get from dad. And those chromosomes, most people can appreciate that the genes there, and there's about 20 to 25,000 genes that build a human organism. And most people appreciate without controversy that they are responsible for our physical structure. Traits that you mentioned, like our eye color, or whether we have curly hair, how tall we are. These sorts of traits are, like I said, not controversial. But when you're talking about more complex phenomena like personality or behavior, this is where people get a little queasy when you bring genes into the picture. And that's not an unfounded concern, because we don't quite understand the aggregate effects of what may be hundreds or even thousands of genes operating in concert to bring about a complex trait, like how aggressive someone is or how prone they are to depression. So I'll give you a couple quick examples that might illuminate how genetics works in the context of various conditions. So you mentioned height. Height's actually a pretty complex trait. There's at least 20, probably more genes associated with height. And there's a strong environmental component with it as well. So the genes that give rise, no pun intended, that give rise to how tall you may be only operate in a certain context. And this is true for most traits. So you can have the genes that in a perfect world would make you 6 feet tall. Okay. But if you are deprived of nutrition or live in an environment where those genes cannot maximize their potential, you're not going to reach 6 feet. And this is true for a lot of cognitive abilities as well. So you have environmental components that are really critical to how genes manifest themselves. You might be familiar with the term nature versus nurture. And this is,
oh yes.
yeah, this is a dichotomy that's been around a long time, where nature refers to the genes, your DNA, and nurture refers to environment. And for the longest time, people on either side have been fighting with one another over which one is more important when it comes to describing what a person is like. And what we found today is that that's a false dichotomy, and we really should never have erected a barrier between nature and nurture, because they truly operate together in ways that I explained for height. So let's talk about a simpler example. We'll talk about sickle cell anemia. This is a devastating disease.
Yeah.
And remarkably, it is caused by a single mutation in just one base pair of billions. Just one base pair that mutation can lead to sickle cell anemia. And, like I said, it's a devastating condition. And if you see that mutation, if you were to sequence somebody's genome, okay, and reveal their DNA sequence, and if you see that mutation, there's 100% chance that individual will get sickle cell anemia. It's incredibly predictive.
Yeah.
But when you see changes in other genes that might be responsible for depression or aggression or some of these other complex phenotypes, it's not so clear cut. So we want to assure people that if we had a crystal ball and could see your genome, actually, we don't need a crystal ball these days. We can see your genome sequence, if you get it, if you get it sequenced. But we want to assure people that the predictive power only exists for a few select genes, such as sickle cell anemia. When you're talking about complex behaviors, we are far from understanding the coordination of multiple genes to get to that behavior.
Another example of this would be certain diseases such as ALS and Huntington's as well, right? There's more of a strict genetic component. I mean, there's other things involved as well, but there's very much a kind of just simple. I don't say simple, but there's a kind of basic just math of adding it up and saying like, look, there's a 50% chance you're going to get Huntington's, based on, there's other certain diseases or disorders or challenges that we can know have a very, very strong genetic component as well. Yes.
You're absolutely right. ALS is a good example. Huntington's is caused by an elongation of a certain genetic sequence, and it's also got a very high predictive index. So, but that's powerful knowledge too, because if you look over the course of history, there are some families who have eradicated the history of Huntington's from their family tree,
right? Yeah.
Through in vitro fertilization and selecting for embryos that lack that insertion in the DNA sequence. And to me, that's just revolutionary. I mean, I think that's just fantastic and a really good example of how genetics can be used in a very positive way, to rid a family tree of a disease that has plagued them for generations.
So you started bringing up nature and nurture. I mean, this stuff goes all the way back to Jean Piaget, who was, a child psychologist and Swiss child psychologist in the 50s and many other folks. But I would agree the dichotomy there is a false one, right? It was, I think it was fine for however many years that it existed because that's all we knew at the time, right? And then as we continue to do science, we start to say, hey, wait a minute, this is not maybe as accurate as we think it is. So there's a couple parts to this question. So what is the role of environment for genes? But how can we understand the interplay between genes and the environment? Because as you're kind of alluding to, many people will get very animated on one side, which is silly, but one of these emphasis or another. So you have many folks that are kind of pushing and encouraging this type of genetic determinism look, this is the genes and this is what it is. And that's it. That's all she wrote. That's going to be your life. Other folks totally ignore the genetic component and say everything's part of an environment. Everything's part of a social construction, et cetera. And it seems that there is definitely an interplay for almost everything between genes and environment. So how do we understand the role of the environment for genes and then the kind of marriage between the two?
Yeah, that's a great question. And you kind of answered it already because there's meritorious arguments on both sides. And as we know, when people are polarized, the truth is always somewhere in the middle. Mm-hmm. And that's exactly what the science is revealing today. So there's a science I'd like to introduce at this point called epigenetics.
Oh, I was going to ask you about that. So good.
Yeah, but it's the science. It's a relatively new science, but that was the compelling evidence that really informed me to that this dichotomy between nature and nurture is false. So epigenetics is a word that literally means beyond the gene. And it's also a bit of a slam to genetic determinists because it unequivocally shows that the environment, while it doesn't change genetic sequence, can indeed change how those genes are activated. And it does this through a variety of mechanisms that I spell out in the book, but we don't have to go into today. But what this boils down to is that the environment can signal your cells to perform chemical modifications to the DNA molecule. Again, we're not talking about a change in sequence like a mutation. We're talking about chemical modifications to the DNA molecule itself. And these are reversible modifications. And they control the activity of genes. They can turn a gene on, they can shut it down, or they can tune it. They can fine tune the expression level of that gene. So when you consider that for the variety of gene networks that we can potentially express, that gives our bodies incredible flexibility in terms of what we would call phenotype, what results from our genome. And it's a little staggering to think about. There's quite a bit of philosophy that trickles in when you read a book like Pleased to Meet Me. And the philosophy in terms of epigenetics is basically boils down to this. There's an analogy I use that your genes are like the piano keys, but the environment plays the song. And if you think about that for a minute, what the implications are is that if you had a clone and that clone, has identical sequence to you, was raised in a completely different environment, imagine the opposite environment that you had, despite the fact that clone is identical to you, gene per gene, there would be a completely different personality associated with that individual. Mm-hmm. There may even be some changes in physical attributes as well as cognitive abilities. So you really can't separate the effect that the environment has on the way genes work. And that's the science of epigenetics.
So I know you talk about epigenetics a bit in the book. I think especially in the beginning part of it. So I'm not completely sold on epigenetics personally in my reading of it for a variety of reasons. So I don't think that it's bad science, but I do think that some folks overestimate the strength of it. And basically my thing is like, well, it's not quite there. I kind of want to do a more wait and see approach. I mean, there's problems with epigenetics, such as, many of the studies that have been done have, moderate effect sizes. They have a variety of cofactors. They have, low replication rates. There's a whole host of issues with some of the epigenetic studies that have been done. What do you want to say about that? Are you going to plant your flag here and be like, no, epigenetics is the way, and or are you also kind of more, I guess, questioning about the long-term efficacy of it, or we need to get more information? Just kind of, I guess, what's your position on some of the criticisms that some folks have had about epigenetics?
Well, sure. As a scientist, I don't plant my flag in anything. Right. I keep an open mind because sure. my beliefs are informed by evidence. And as additional data comes in, I will adjust my conclusions accordingly. Mm-hmm. I have no ego about any of that. And I don't think anybody should. I think this is one of the dangers that we have. One of the shortcomings humans have in building a society is we're too rigid. We're too proud and vain, and we should be open more to uncertainty and not feel bad if we want to change what we think or what we believe in as new evidence comes in. In fact, you could argue that it's kind of insane not to. Mm-hmm. So with that kind of preface, what I do get excited about is that epigenetics, at least in the animal models that I have seen, mm-hmm. Some of these studies are highly reproducible. And when you see the same phenomenon occurring, whether you look in fruit flies or roundworms or mice, when you keep seeing the same phenomenon popping up again and again and again across very different species, that's a good indicator that you're onto something real. Now, how this translates into humans is another ball game entirely, because we can't do those sorts of controlled studies across generations as we can for these, insect and animal models. So the best we can do are try to make correlations based on historical data or indirect evidence, of which there's some pretty compelling data, but you do have to take it with a grain of salt. You're absolutely right about that. And epigenetics is a new science. It's really only been around for 20, 25 years. And as you alluded to, quite correctly, people get a little carried away when some of these studies come out, and they might want to jump to conclusions. We see the same thing with studies about the microbiome, which is also touched on in the book. And in fact, I put in a couple of disclaimers, I think, near the end, where we talked a lot about epigenetics. We talked a lot about the power of the microbiome, but for the very reasons that you mentioned earlier, we got to be cautious. We can be excited about the possibilities, but we need reproducibility, and we need more research, greater enhancement of the data before we draw firm conclusions.
Yeah, it reminds me of the kind of excitement that people have about GWAS studies and some of the SNPs and all these things. And again, there's a lot of really good potential. And I think in certain isolated studies, it is pretty powerful, but it is not in others. And so there's not this universality that people kind of want. And so, I will tell folks that, they'll share, a GWAS study. I'm like, yeah, I mean, I see that. But again, here's some of the many issues with this as well. Maybe let's not, start throwing the streamers and celebrating about all of these things. Like it's not necessarily saying that. It has potential. I think, this is very much in the weeds, but there are some scientists that will say like, well, we should, for particular things, GWAS studies don't really tell us anything. And so why would we do that research anyways? And other people will say, well, of course we should do it because there's a lot of potential and we don't know what it's going to keep telling us in the future. And, people fight about that. But I think that it's, kind of the main point is the same that you're making here with the epigenetics, which is broadly is, how do we make sure that we are very optimistic, but also very honest about what we're doing, whether it's particulars with GWAS studies or whatever else, or just this kind of broad kind of field of epigenetics overall. So I think it's a good point that you mentioned.
Yeah, there are some parallels there. Personally, I'm a fan of the GWAS studies. I do agree they certainly have limitations and we have to be careful how they're interpreted.
Yeah.
And for some traits, they're more informative than others. But some of the GWAS studies that I have written about and researched myself are pretty compelling in regard to like the polygenic index that they generate. They can be just as predictive as some of those socioeconomic factors that social scientists point to in terms of, things like IQ or, standardized test scores and things like that. So they do have utility. I certainly wouldn't want to be in a world where they're ignored and dismissed and not analyzed. Because even at the end of the day, I mean, they're getting cheaper and easier to do. So the data is there for the taking. Why not at least examine it and see if there's some strong correlations between certain genes or gene networks and outcomes for people. And it depends what outcomes you're looking at, of course. People get a little queasy, when you're talking about intellectual ability. Take IQ, for example, which is just a small slice of the intellectual pie, in my opinion. It's just one small facet of what we would consider intelligence. But some of the polygenic scores are really good for IQ performance.
Well, on that in particular, I've talked to a handful of people that when they look in their crystal ball, they'll say, look, it's not unfathomable to think that a polygenetic index score is going to be much more accurate than an IQ test or certain aspects of IQ. And that's a potential. And I think that potential is, it's just going to make the current debate that people have on steroids, right? We're going to need an ethics on how we have a clear understanding and, kind of a human way of trying to explain polygenetic index scores and things like that.
Yeah.
We'll see where the future goes, but I definitely think we'll need some type of ethics whenever we get there to that point. It's very possible, but yeah.
Yeah, the ethics issue is critical. And a lot of times our technology flies far ahead before we think about the ramifications of what it might mean. And of course, when you're talking about behavioral genetics, it's always tainted with the horrible history of eugenics.
Right.
But that's not the intention of what behavioral geneticists are trying to achieve today. They're actually trying to improve and better human lives. Yeah. And the ethical question that comes to my mind is, we have the power, we have the ability to identify subsets of the population who may benefit from additional tutelage in a certain area, spatial math, for example, because they have a polygenic index that suggests they're going to be a little deficient in this area. To me, that's very useful knowledge. And if it was my child, I would like to know that piece of knowledge so that we can compensate for that potential deficiency. And on the flip side, if they have a polygenic index that suggests they're going to excel at a certain activity, I think that is also very useful to know.
Yeah, I absolutely agree. And I totally agree with your position there on a lot of the times our technological advancements are sometimes light years ahead of our ethics. And, we have to kind of fix a crooked line at some point. So, I totally agree. So let's get into some of the specifics. You walk through the book talking about different things: taste, appetite, mood, and so all these different things we'll cover. But you start with taste, which is super cool. You talk about some of the genes, the TAS2R, and the SCLA2 and the GLUT2. And so there's a lot of genes implicated here. So maybe kind of just, march us through some of the big ones and how they're important and how these genes and maybe others are involved in taste, and then maybe some of the other senses.
Yeah, this is a fascinating subject, and it's one that I think interests a lot of people because as social creatures, we're always talking to each other. You got to try this food. You got to try this drink, and they might not like it as much as you do, and you're baffled by this. You're like, well, you got the same kind of tongue that I do. Why don't you like this? What's wrong with you? And this is a personal story for me, and it was one of the reasons I was motivated to write this book, and particularly that chapter, because I happen to be what scientists refer to as a super taster. Okay. It goes to this gene you called, the taster gene, the TASTR. And I got myself genetically tested because all of my life, I could not stand vegetables like broccoli. They just tasted awful to me. And most people I knew were tolerant of this vegetable. In fact, they actually enjoyed it. And I couldn't figure this out. I was, this is disgusting. And to this day, if my wife, who loves broccoli, if she's cooking in the kitchen, I have to leave. I can't even inhale the vapors from it. It just makes me gag. And it turns out I have a genetic variation in that taster gene. And the way this works is the taster gene builds a protein. It's a taste bud receptor. It's on my tongue. And what happens is, in most people who have a normal copy of this gene, the bitter chemicals in broccoli and other cruciferous vegetables just kind of float right across that receptor. It kind of just gives it a fist bump. Okay. But I have a mutant taste bud receptor. And when they encounter those chemicals, they give it a bear hug. They glom onto it really tightly and don't let go quickly. And that signals to my brain that, dude, you just ate something poisonous. You better spit it out. It's too bitter. It's not edible. Get rid of it. And that happens in about 25% of people. So we are a minority. And that's why when I was growing up, not too many other people were like me. But as I got older and, researched this a little bit, got myself genetically tested, I am a bona fide super taster. I have this mutation. And I was vindicated in a way. I was like, this is not my fault.
Mm-hmm.
I genuinely can't tolerate broccoli. My brain thinks it's a poison. And there's an evolutionary explanation for that I can expand on if you like. But at the end of the day, as vindicated as I felt, it's a little unsettling at the same time, because what this implies, if you think about it, is that we're really not in control of some of the things we like or don't like.
Mm-hmm.
This is governed, at least in part, by our genetic makeup.
Well, I was curious about the evolutionary piece there, because obviously the first thing that comes to my mind is the role of disgust. So when you look at the role of disgust, kind of your basic disgust is, pathogen avoidance. And I wonder where that kind of, disgust is kind of seen as a system. Where does that kind of physiologically fit into here of, your body's, based on the genetic variant, is telling you this is something to resist because it's, something that is, just abhorrent to you. And so I wonder, it seems like broccoli is very good for you, I think, right? It's a good vegetable for you. It's nutritious.
No question about it. And that's why I wanted to like it, Xavier. I wanted to like it because I like healthy foods, but my body just wouldn't let me. So here's the evolutionary theory, which I find fascinating.
Yeah.
There's no proof of this. We can't test it without a time machine.
Sure.
But the theory makes some intuitive sense. So natural selection experiments, because you don't want your species to be homogeneous. You want variety within the species. Yeah. One of the most intuitive examples is immunology. We have different immune systems and we respond differently to pathogens, so that if something comes along that wipes out most people, there's going to be a fraction of the species that survive because they have a different immune system that's governed by genetics. So this phenomenon of tasting, how that fits into this context is that plants, they're manipulative creatures. Okay. They don't want to be eaten, but we eat them, right? So they engineer chemicals within the plant that make them unpalatable. Okay. And broccoli has that to some extent. Okay. To the super tasters like myself, 25% of people are going to find them unpalatable and they're not going to eat them. Whereas 75% of people will think they're fine and eat them. They will reap a nutritional benefit and I will lose out on that nutritional benefit. Fortunately, I can look to other foods. But back when we were, just climbing down from the trees, if we had a tribe that moved into a new area and started consuming vegetation that was unfamiliar to us, the super tasters might have not eaten a plant that did turn out to be poisonous. So the 75% of people who gorged on it might have become sick, possibly even died, whereas the super tasters would have lived and carried on the species. So that is the evolutionary model for why super tasters might exist.
Yeah, it's interesting. Obviously, we can't prove it, but you're correct. It does make this type of intuitive sense about how this could be of why these certain people have this gene and a variant and others don't. I guess moving on to, we'll get to appetite, but obviously something that I was reading with much interest, and I think many of my listeners and folks that I know will also find this interesting, is the role that caffeine plays with our genetics and with people in general. So maybe tell us kind of a little bit the story you tell in the book about caffeine. It's a very, 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 very highly socialized stimulant. I definitely drink way too much of it. And so explain to us how caffeine works, I guess, genetically and in our body.
Yeah. One of the things I found out back in my blogging days was if I wanted to get a lot of hits, a lot of readers, all I had to do was write about caffeine or coffee.
Yep.
And people would flock to it. So it's obviously a very near and dear drug to a lot of people, myself included. I'm just like you. I really don't have much of a personality without a cup of coffee. But
I definitely need it in the morning. I will get the headaches. I'll have the withdrawals. I sometimes don't need it in the afternoon, and I will drink it in the afternoon, but definitely in the morning. I can't. I'm not functional.
Yeah. And you're not alone. I think that's probably describes the vast majority of at least Americans. So here's what's going on. When we wake up in the morning, we have very low levels of a chemical called adenosine coursing through our veins. And as the day goes on and as we stay awake, this adenosine chemical builds up over time. And it's basically a signal to our brain that tells the brain how long we've been up. Okay, it's like a clock of sorts. It's a chemical clock. And once we accumulate a certain level of adenosine, it binds to adenosine receptors in the brain and signals to the brain that it's probably time to start winding down and go to sleep. Okay, makes you tired. So structurally, if you look at the chemical structures of caffeine and adenosine, they're very similar, which means caffeine, when it gets into your body, goes to the brain and it sits in the receptor. It sits in that chair where adenosine usually sits. Okay. So if caffeine is sitting in adenosine's seat, adenosine can't signal to the brain that you're tired. So your brain is fooled into thinking that you haven't been up that long and it's not time for sleep. So it's, pretty neat little hijacking of our natural sleep and wake cycle. So the problem, as most caffeine consumers know, is that the body tries to fight this. It figures out what's going on. And after a while, it, starts to express more adenosine receptors. And the effect that has is it makes us drink more caffeine because the 1 cup doesn't cut it anymore. So we increase it to 2, and then we have to take 3 just to feel the same effects of 1 cup. It's the signature of drug addiction, right? Building up a tolerance to drugs. And caffeine is a drug. It's minor compared to some of the other ones that we talk about in the book, but it operates in the same fundamental way in terms of our body building up a tolerance and trying to make adjustments. So what I've tried to do over time to manage my caffeine obsession was to try to drink it strategically. I cut it out of my mornings because I hated waking up with a headache and hated waking up having to go straight to the coffee maker in order just to get ready for the day. I'd need that hit to wake me up. So I stopped trying to drink it and require it in the morning and try to reserve utilizing caffeine only when I need it most, like when I'm going on an exciting podcast. You drink it before that podcast and you experience a greater level of alertness. It does more. So you basically want to try to strategize how you can use caffeine to your advantage. And if you're using it constantly all the time, I don't think you're going to be able to reap the benefits of using it more strategically.
I think it was Poland, right? He's a very popular writer who talked about how he took, was it a month or 3 months? I'm going to get it wrong. Off of caffeine entirely, cold turkey, just didn't have it. And he said that first time he had a cup of coffee or whatever, there's a really nice latte or Americano or whatever it was, after not drinking it for, I don't remember if it was a month or 3 months. It might've been 3 months. He said it felt like it was like a hit of cocaine or something. It was just so potent. He just hadn't had it, for a while. And he was a regular coffee drinker. And it's interesting how that works, right? How that works in the body, how there's this kind of deception that occurs. And it's we need more and more and more. And that's true for many substances. I guess since we're on substances, we can talk about that here. You tell a really fascinating story about, Ozzy Osbourne, which I kind of heard something of that, and I thought it was kind of like a myth, but that he has this genetic tolerance for substances. Tell us about that story. You can use his example, I guess, of how some people have a stronger tolerance for alcohol or maybe even other substances, and how that kind of works.
Yeah, it's a remarkable story, and most people haven't heard about it. So I think it was 10 years ago, maybe back in 2009, Ozzy Osbourne, who I don't think needs any introduction here. Ozzy Osbourne,
King of Darkness,
the Prince of Darkness, right? Was one of the very first people on the planet to get his genome sequenced, and it wasn't his choice. He obviously volunteered to do it, but scientists actively sought him out to be one of the very first people to get their genome sequenced, and that might cause some people to scratch their head. Why would they want Ozzy Osbourne? You got all these great political leaders, all these intellectual giants, athletes. Why not go after them? See what makes their genome tick. But no, they went after Ozzy Osbourne because of his reputation for being able to put away ungodly amounts of alcohol, just crazy amounts of alcohol. And they thought that sequencing his genome might reveal something about why he was able to tolerate so much, and maybe why he struggled with addiction, with the aim of ultimately helping people who struggle with alcohol addiction. So Ozzy Osbourne was game to do this, even though he admitted the only Gene he ever knew anything about was the guy from Kiss, right. Gene Simmons. Right. So, typical sense of humor that Ozzy has. But when scientists sequenced Ozzy Osbourne's genome, they found a never before seen mutation in a gene called ADH4. I think that's what the gene was. It stands for alcohol dehydrogenase. And this enzyme functions in the liver, and it detoxifies alcohol. And the nature of the mutation that Ozzy Osbourne had would have meant that he is expressing just tons and tons of this enzyme. So his liver is like operating in fifth gear, and it potentially explains, I don't know if anyone's done studies to confirm this, but it potentially explains why he could tolerate so much more alcohol than the average person. It's because his liver's processing it so quickly. Now, where this gets interesting in terms of potential treatments is that many other GWAS studies have been done on people who have struggled with alcohol. And, this is a great selling point for the value of these genome-wide association studies and their link to behavior. And some of these studies were actually done at my university, at Indiana University.
That's really cool.
So they basically just sequence the genomes of many people who struggle with alcohol, and you sequence like a similar number of people who don't struggle with alcohol, and then you look for the differences. Pretty common sense experiment. And what keeps coming up time and time again in these studies are aberrations in a neurotransmitter pathway that's regulated by GABA. So for those who don't know, GABA is a neurotransmitter that calms the brain down. And there's many genes that control this. There's genes that build GABA, genes that break it down, and of course, genes that build receptors that bind to GABA. So there can be many different ways this pathway can be affected by genetics. But at the end of the day, what this means is that people who are prone to alcoholism typically have an overactive brain. They lack sufficient GABA, this neurotransmitter that calms the brain down. So these individuals tend to have trouble focusing. They tend to have ADHD. They have trouble sleeping at night because their brain is just firing all the time. I can't imagine how torturous that might be. Yeah. But I actually met someone on the book tour who struggled with this very problem. And there are medicines now that are non-addictive that can help calm the brain down so that these individuals can focus and sleep better. But what normally happens is that someone with a hyperactive brain, probably in adolescence, experiments with alcohol and finds that it calms their brain down and it helps them sleep, and to a certain degree helps them focus, makes them feel normal. So it's almost like a medicine to them, but it's a slippery slope because it's a highly addictive substance. And like we talked about for caffeine, the more alcohol you consume, the more you're going to need in order to feel the same effect. So that's where we get into a really dangerous problem. So to summarize that information and what the power of those GWAS studies is pointing to is we can now identify tangible biological roots to the alcoholism problem and design non-addictive medications that help people with this GABA deficiency in the brain.
Yeah, I think that's what's really, really important is you're kind of, you are kind of tying it all together of some of the genetic studies, and then how we can have better treatments, and so people are not having these pretty serious addiction problems. Let's move to, I guess, appetite, because I don't want to skip that. Just in general, we can hit a few points here. First, what are the ways in which our appetites work, and how we've decided what a healthy caloric intake is? You mentioned the gut microbiome, which is something that people have become more and more interested in. How does that connect with appetite? So just tell, and some of the genes that are involved with appetite in general for humans.
This is a huge subject, and that's why I dedicated an entire chapter to it in the book. Mm-hmm. So metabolism is highly complex, and even though from like a simple understanding, you think you eat, you get full, you stop. That is a highly regulated system at a molecular level in the body, and there's many ways it can be broken and things can go wrong. So you mentioned a gene earlier, for example, this SLC. It encodes a protein called, I think, GLUT4. Mm-hmm. So this is a gene that varies in people, and it's basically a sweet tooth gene. So some people have a variation in this gene that builds this glucose receptor. So it binds sugar, and the variation disrupts that ability. So this basically a sugar sensor in the body has been broken, and the brain can no longer assess how much sugar is in the bloodstream. So people keep eating, because their brain hasn't gotten the signal that, okay, we've had enough calories, we're done, it's time to stop eating. That signal pathway's been disrupted. So, and that's just one example. The other example is leptin. Okay, this happens in about 10% of people who are obese, but it's yet another instance where a genetic variation leads to a disruption in the brain's ability to feel the sensation of a full stomach. So people tend to overeat because they're not getting the signal that they've had enough.
Interesting.
And you can recapitulate these in mice. That's the most remarkable part. If you put this same leptin mutation into a mouse, it will start to overeat and even develop diabetes.
What is it about that's just terribly fascinating. What is it about the microbiome that is so captivating for people, in which they are looking at this and saying, like, there's more bacteria in our body than there is other things. There's so much importance with it, how it connects with the brain. There's been quite a fascination with this. What's the involvement there for our kind of body?
Yeah, this is one of the studies that literally almost knocked me out of my chair. It's so crazy, but it's been reproduced many times. So I don't doubt the validity of it. But let's talk about the microbiome for a moment. I don't know how many times you might have talked about that on your podcast in the past, but this is what we're referring to are the trillions of microbes that are inside or on your body. And when we're talking in the context of appetite and obesity, we're talking about your intestinal microbiome. You have hundreds of different species of bacteria that populate the inside of your gut, the intestines and the colon primarily. And they've been known to exist for a long time. And they've been known to be important for breaking down certain foods. Okay, fibers, for example. They help us digest things. They make certain vitamins. So they have some certain health benefits. But it's been hypothesized a while back that they do much more, because these bacteria, just by virtue of growing and replicating, are producing tons of biochemicals that may be interacting with our body. And there's evidence that they even interact with our brain. They can travel up the vagus nerve, connecting what is going on in the gut with what is going on in the mind. So fascinating stuff. So the key experiment that knocked me out of my chair was done in germ-free mice. So this is a valuable tool to study the microbiome. So germ-free mice are born through a sterile C-section, and then they're kept in cages where they're not exposed to any microbes whatsoever. So they literally have no bacteria growing on them or inside of them, which gives scientists a tool to then transplant bacteria from another source into that mouse to see what happens. So what happens is there are identical twins in humans that are discordant for obesity, which means one of the identical twins is lean and the other identical twin is obese. And this is what got people thinking there might be something more than genetics to the obesity equation, because identical twins have the exact same genome. How in the world can one be thin and the other obese? Good question. Maybe it's the microbiome. So what scientists did was they harvested intestinal bacteria from the lean twin and put it into one of these germ-free mice that I told you about. And essentially nothing happens. The mouse stays lean and healthy. But when they take intestinal bacteria from the obese twin and put it into that same germ-free mouse, the mouse starts to overeat and become obese. It's just a mind-blowing experiment. And what this tells us at the end of the day is that there is, in fact, evidence for a microbiome component to the obesity problem.
It's super, super important, because when you think about how long that's been the case and we haven't known it, right? It's just one of these wonders of science. And I guess what are the I mean, as far as you know, what are the current, processes that are happening in science that are trying to rectify some of the challenges that this presents for people, and to say, oh, well, we know some things about this now. Well, now we can correct for some of this stuff. So, I mean, I don't know how close you are to this literature, but what are some of the ideas that are kind of being thrown around about how, now knowing it, how we can then have preventative or proactive measures for folks?
No, great question. I think there's two main takeaways from these types of experiments, and we'll circle back to addiction as well, because that's got some parallels with the obesity problem and how society historically has perceived these issues. So historically, things like addiction and obesity have been in the realm of willpower, discipline, and it's pretty much on you to put down the bottle or put down the fork. And that's never really been a very edifying explanation. Okay. It just raises another question. Why can some people do that, but other people can't? And what we are now understanding through these types of experiments is that there's elements that we don't have control over, whether it be genetics or the microbiome, that are basically making it more evident that things like addiction and obesity are bona fide diseases. They are medical conditions that we can now do something about them. Once we understand the biological nuts and bolts of their origins, we're in a better position to solve the problem. I already mentioned the medications that can substitute for GABA in the case of addiction. So what can we do in terms of obesity and the microbiome? Well, scientists and many pharmaceutical companies are intensely investigating that possibility, because the microbiome can be replaced or remodeled. So some of the, they're not very glamorous names, but there's something called a fecal transplant, which some people have received to treat another medical condition, not obesity yet. But we can do these in mice and they work really well. So the proof of concept is there. But to the best of my knowledge, there might be human clinical trials underway, but I'm not privy to the results of them so far. But in mouse models, when we manipulate the intestinal microbiota, we can change metabolism and appetite and weight. So it's pretty promising stuff that we can basically, we're at a point now where we can envision some sort of probiotic pill that's going to contain the magical bacteria species, or maybe even what they make, what metabolites they generate, as a treatment for the obesity problem, to give these individuals the biological assistance that they need to overcome these problems.
Yeah.
And hopefully in the process, destroy the stigma that society has attached to people who struggle with these issues. Rather than shame and blame, let's roll up our sleeves and try to figure out what's wrong so we can remedy the problem.
Yeah, no, absolutely. I think the potential is there, and I think it's super, super important. And 10, 15, 20 years from now, I think it's only positive and very promising of what can happen. So
I hope so too.
Let's move to emotion states. Is a middle part of the book, I guess you could say, on mood and relationships. And so we can talk a little bit about various mental disorders. There are many neurotransmitters that are involved with mood, such as dopamine, serotonin, norepinephrine, and also cortisol is hormones is involved as well. What's the kind of overview on these neurotransmitters and how they're involved with various mood disorders? And then we can, talk about some of the genetic components of these mood disorders as well.
Yeah, I think a great way to approach the answer to that question is to walk through three of the hidden forces that we've already talked about today: genes, epigenetics, and the microbiome. So we'll start with genes. As you already alluded to, our moods are basically governed by complex interaction of neurotransmitters in the brain. Serotonin, for example, needs to be at just the right concentration. Too much is bad for you. Too little is bad for you. Mm-hmm. And this is exactly the neurotransmitter that's targeted by antidepressants like Prozac. Right. So we've been gaining a better understanding of depression and why people just can't, quote, snap out of it. It's because they simply don't have the neurochemistry that normal people possess. Right. And that is just a misfortune of fate. It's certainly not their fault. Mm-hmm. And some of this has genetic origins. Okay, so that's one component. The second one is epigenetics, which has been more recently appreciated. So there's a fascinating study that's been done in humans that looks at children who have had adverse childhood experiences, or ACEs, as it's known in psychology. Mm-hmm. So these unfortunate children are subject to abuse, neglect, or bullying throughout most of their childhood or all of their childhood. And it's long been appreciated that such children possess psychological scars that wound them in ways that when they become adults, they struggle with stress management. They have high anxiety. They're more prone to aggression and suicide. And people just usually assume that that's a psychological, repercussion of the horrible environment they've experienced. And there's nothing wrong with that answer. But as a scientist, I want to know the molecular details of what's going on. Okay, why do they have a psychological issue in these realms? And epigenetics is revealing potentially why. So how this works, just real briefly, is that scientists have basically done postmortem analysis of children who have had adverse childhood experiences and then sadly committed suicide later in life. Mm-hmm. And when they examine the neurons, the brain cells of these individuals, they find an enrichment of epigenetic modifications at stress response genes. And what this ultimately means at the end of the day is that the genes that normally help people regulate stress, and they relate to the cortisol that you mentioned earlier, and the cortisol receptor that's responsible for cleaning out the system, they have epigenetic modifications that dysregulate the expression of those stress management genes. And that basically results in an inability to regulate stress. There's high anxiety. And this explains many of the unfortunate issues that these children experience as adults, which we formerly characterized just as psychological problems. But now we're finding that these are actually scars on their DNA, epigenetic scars. And with the future of how this can be powerful knowledge, in the future, we're hoping to design drugs that can rearrange those epigenetic marks. And there's been some encouraging progress in animal models, again, where scientists are designing drugs that can reverse these epigenetic changes and perhaps save these children from these horrible outcomes, even though they've unfortunately experienced adverse childhood conditions early in life. And then finally, there's the microbiome. So going back to the germ-free mice that we mentioned, I should preface this by saying mice are just like people. They're pretty happy creatures. They're social, okay? And they enjoy treats and things like that. So if you take one of those happy-go-lucky mice and transfer intestinal bacteria from a depressed person, and you put it into that mouse, it starts to exhibit symptoms of depression. Remarkably, those mice lose interest in treats. They don't socialize with their buddies anymore. Wow. And if you throw it into a bucket of water, it won't even try to swim out. It won't try to save itself. So this, again, one of these mind-blowing experiments where the unappreciated effect of the microbiome could actually be reaching into the realm of depression and potentially giving us a new way to treat it.
Yeah, that's super fascinating. I didn't know that last part. It's one of these things where for a long time, people have had certain ideas about these things, but now we're seeing with various disciplines looking at this stuff, how interconnected the human body is. I mean, I think we can probably all know that and say that, and of course, but really understanding the systems that are at play of what's connecting all of these things. And therefore, the treatment of it is going to be much more multifaceted, right? It's not, here's some Prozac and, I'll see you next week or whatever, see you next month, right? There's a whole aspect of, exercise, diet, how you're able to, do, I think, elements of therapy, how you're able to use psychotropic medications, et cetera, and other aspects of life. So I think it's really telling how all of this is really included.
I'm glad you raised that point, because that is one of the major themes of the book. There's rarely a one size fits all when you're talking about complex behaviors or personality traits. It involves genes, epigenetics, maybe the microbiome. And just a nice quick example of how it all fits together, we'll go back to the microbiome and depression. Okay. The microbiome, the bacteria in your gut actually make 90% of your serotonin, which is basically puts a little more explanatory power into why the microbiome could possibly be connected to depression. Right. It's because these bacteria, certain species of them, make the very neurotransmitters that govern our mood. And something else they make? They make a compound called butyrate, which is an epigenetic drug that scientists are working with today. So these bacteria literally have the power to modulate the activity of your genes. And we're just beginning to understand how all of this works. Yeah. I'd like to touch on the very important point that you raised. You talked about proper diet and exercise. A lot of studies, as boring as that advice sounds, a lot of studies show that if you have a diet of processed foods that's really high in sugar, fat, and salt, and if you have a lack of exercise, this really screws you up big time. And the way that it does is not necessarily through genes, but it's through the epigenetics. Okay. Exercise has been shown to modify the epigenetic marks at many gene networks associated with handling stress, managing appetite, and increasing intelligence and learning. And of course, the microbiome is profoundly affected by someone's diet. And we now know that you screw around with the bacteria in your gut, they're going to be making a different array of biochemicals that could potentially explain a lot of the strange problems that highly developed societies like the United States have. You know, we're pretty unique in having a lot of people with allergies, a lot of people with depression, a lot of people who are obese. And it's premature to say it, but what if all of this relates to the diet and exercise that we have in this country, which was just pretty horrific and very against the grain in terms of what we were evolutionarily designed to consume. I think, you know, it's probably non-controversial to say that many of the maladies we face could be resolved by proper diet and exercise.
Yeah, I wholeheartedly agree on that. And I do think that we, you know, have a, you know, in this country, a terrible relationship with food for the most part, and or, you know, with diet. I think it's, you know, anytime I've traveled abroad, you know, it's just the quality of the food is usually different and better, and, you know, smaller, and, you know, it's just, it's a big difference. And I think that's, you know, slowly having a pretty big impact on many components, as we're seeing. It's not just like, oh, what's my GI tract doing, right? It's more of like, oh, like, how is this impacting mood, and how is this impacting brain states? And so I think that's, you're right. It's not very sexy advice, but I mean, it is something that is very true, though, and how it's, you know, integrated with so many other parts. I want to just touch on briefly here on aggression. You mentioned it in the middle part of the book. You talk about the MAOA gene variant. Serotonin comes into play here. So, and then also there's the role of environment and childhood experiences. So maybe just talk about all those kind of different angles with aggression.
Sure. Again, there's a whole chapter dedicated to this because there's a lot of exciting science coming out, some more controversial than others.
Yeah.
Now, we'll discuss MAOA in particular because it's one of the better characterized, better studied genes that has been implicated in the loss of impulse control and aggressive behavior. So in fact, MAOA was the first gene that was used in a court case back in 2009. And this is when history was made. It basically exonerated someone who murdered another person. And instead of getting the death penalty, the person just got imprisoned for life because his lawyer successfully argued that because he has a particular genetic mutation in the MAOA gene, he was not, quote, fully responsible for his actions. Mm-hmm. So let's try to make some sense out of all of this and see where it stands. So MAOA is a gene that builds a protein called mono A oxidase, mm-hmm. and it functions in the brain to regulate the amount of neurotransmitters that are coursing through the brain and influencing people's thoughts and impulse control. So when this particular mutation occurs, and this has been shown in animal models as well, in mice, there's a particular mutation that basically results in a monooxidase gene that doesn't function very well. And it leads to a dysregulation of neurotransmitters in the brain that are correlated with aggressive behavior. So this gene got a lot of intense research attached to it early on. And like I said, if you recapitulate this genetic modification in mice, those mice stop being those friendly social creatures and become ferocious. And there's other examples of genes that can be modified in mouse models that do the same thing. So there does seem to be a genetic component to violence and aggression. But where things get interesting is that it doesn't happen universally. So there's people who have that same mutation in MAOA who would not hurt a fly. Now, this is puzzling, so it goes back to this point that you raised earlier, when we're talking about complex behaviors, it's usually not a one size fits all thing. Mm-hmm. It's a complex equation and there's lots of moving parts.
Yeah.
So there's something we're missing about MAOA that we have yet to discover that might explain why it does one thing in some people and it doesn't in others. Mm. So to date, what's been known so far is that the environment comes into play. Mm. So if someone has the MAOA mutation, is the so-called warrior gene mutation that is connected with aggression, and they've been abused as a child, they have a very high predisposition to being aggressive and violent. Whereas someone with the mutation who is not subjected to adverse childhood experiences will be just fine. Hmm.
It's interesting how I'm thinking about, people still argue about what a gene is. And so they'll say, is it the material or the information? People have disagreements about that. When you were talking earlier, my knowledge is that we have 3 billion base pairs with genes, but only 20,000 are working to make protein. So then the question is, what are the 2,900,080 whatever? What are they doing? They can't all just be ancillary. And so there's still like a whole universe of things to understand about genes and how they work. And I can't help but imagine that there's certainly, as you're describing, a genetic component with aggression. I'm thinking of the I'm going to forget the study. There's that study with the different types of they're not deer. What is it? With the antlers? Oh God, I'm going to forget it. Where you look at aggression, you can say that it's definitely a genetic component, but then you have to also look at it with the environmental piece. That there's just all of these variables that are occurring with aggression in other animals and then also in humans. And so I think it can be, this has real world implications, right? As you gave your example with someone killed someone, I can have the moral, argument later about whether the death penalty is correct or not. But, still, this has, someone died, obviously, and then someone is serving life. And there's like elements of that. And this gets into testosterone, and this gets into some of the gender stuff that we've seen before about are men more aggressive than women, et cetera, et cetera, et cetera. So there's just all this stuff at play with aggression, which is interesting because it seems that it's always going to be complicated. Is the short version. This is very complicated. Is there any other components about aggression that you want to highlight here?
Yeah, I'll talk about the sex difference. I find that fascinating too. It's no secret that men tend to be much more aggressive, fly off the handle more easily, and their aggression usually comes out more as physical violence.
Right.
So we'll circle back to MAOA real quick. That's a X-linked trait, which means that gene is on the X chromosome. Mm. Okay. So most people know females are XX. They have 2 copies of the X chromosome. Men only have 1 copy because they're XY.
Right.
So what this means is that women get 2 copies of MAOA. So if one is broken, they got a backup. Mm-hmm. Men don't. They only got the 1 copy. So if it's broken and they're in that poor environment, that might explain, at least in part, why men tend to be more aggressive and violent, because they don't have that backup system that women have. So I thought that was another interesting spin on the genetics of MAOA and maybe the sex differences in physical violence. You also mentioned the vast amounts of DNA that don't seem to be connected to a gene. They don't contain a protein coding sequence. Right. And we refer to that in my field as the dark matter of DNA. Some people call it junk DNA, which I think is a really big time misnomer, because just because you don't know the function of something certainly doesn't mean it's junk. Right. And rarely is biology wasteful. Mm-hmm. Biology isn't going to keep coding these billions of base pairs that, for no reason. There's got to be something. So and this brings us back to epigenetics. A lot of these the dark matter of DNA is epigenetically modified. So what people are thinking is that these regions that don't encode for a protein regulate that gene. And that explains a large percentage of the so-called dark matter of DNA, these sequences that don't seem to be linked to a protein coding sequence. They're probably still important at an epigenetic level. The clearest example of that comes to my mind is Fragile X syndrome. This is a disease that causes profound cognitive disabilities and behavioral problems, and it's been linked to autism as well. But Fragile X syndrome is an epigenetic disease. It basically is an expansion of a non-coding sequence that is upstream of a gene that's really important for brain development. But what happens is this sequence gets expanded, and then it becomes prone to an epigenetic modification known as DNA methylation, and it shuts that gene off. That's really important for proper brain development. So it's at the end of the day, it's telling us that the dark matter of DNA really does have an important function, and at least part of it is regulating the activity of genes.
Yeah, it just makes, I think, things very exciting for folks trying to still do this research and figure out what else is going on there. So, and the third part of the book, I would say, you talk about mental state a lot. You talk about IQ. You talk about cognitive biases and beliefs. So we've touched on IQ a little bit. I've talked about IQ on the podcast before. So we can maybe just kind of wave to it a little bit. But I guess what I'm curious about is, what are the parts of the brain that you mentioned this and I forget maybe 8th or 9th chapter, I don't remember, of that are involved in creating, shaping, and maintaining beliefs. What is that role the brain has at the genetic level or just at the kind of neuroanatomy of how we understand how beliefs are, made and shaped and continued?
Yeah. That's a great question. And there's no complete answers out there because the brain is kind of the final frontier. It's the organ that is most important, but the least understood. So this is one of the things I'm looking forward to as a scientist to follow up on in the future is seeing how brain science unfolds. We're getting better and better tools, and we're also understanding better how individual genes build and regulate the neurochemistry that courses through the brain. And of course, genes aren't everything because the brain it takes on kind of a life of its own after it's built, and it is susceptible to things like age or injury or tumors or things like that. But basically, to address the main question there is that certain brain areas differ in size, usually in very small ways, but at a molecular level, those could be profoundly important. And there's one component of the brain in particular, it's called the amygdala. And it's basically the emotion center of the brain, particularly important in the fear response. And what some political scientists claim in studies that I don't think have been reproduced where I'm, fully confident in the results, is that they argue that in like conservatives, the amygdala is bigger. And that would imply a heightened fear response or a heightened ability to detect threats. Which you can fathom to be an evolutionary benefit, right?
Yeah.
In some instances.
Yeah. Sorry, I don't want to cut you off. I just want to jump in here. So I've read some of the papers in neuroscience. And so size does not necessarily equate more in terms of ability or quantity even. A very simple elementary way of explaining this is you could have a smaller sized amygdala, let's say, which is kind of the warehouse or seat of emotions, but you could just pack a lot the same or more. The size doesn't really tell you. The only thing it's really telling you is that there is a difference in the shape of it and the size of it. It could have in terms of functionality, more, less, or the same. And so it doesn't really, in terms of the production of things, doesn't really, signal that, too significantly. People make the same arguments here, again, about humans are sexually dimorphic, right? So, there's difference, but, men and women on average have basically 50-50, same types of intelligence, and there's some variance with academics, but the size of their brain or certain parts of their brain is bigger for women. But that doesn't necessarily mean they're smarter or they're more educated so I think, and again, this isn't applied for every aspect in the brain, but I just want to kind of sneak that in there about some people would walk away with, oh, well, this is bigger and it's a better size, and so they must have, more capacity for this. And that doesn't necessarily equate that.
No, that's a really important caveat. And if I recall correctly, some of the criticisms of those types of studies, and this goes for MRI studies too, just because you see a heightened amount of glucose rushing to a certain area of the brain doesn't necessarily mean it's got a greater function, or that, that part of the brain is more important than other parts in whatever aspect of physiology they're studying. So those are indeed very important caveats. And we know so little about the brain. We got to be careful in the interpretation, because you can imagine that even though one has a larger amygdala, what really matters is what sort of proteins are being expressed in that amygdala and what sort of neurochemistry is flowing through it, not necessarily the size, right? So in response to some...
I'll make one more point here on that. Is also the same is true, or not the same, but there's an element here with fMRI studies as well. Right now, fMRI studies are great. They can, it was a good advancement with the technology, but because of what we know about brain anatomy and functionality, because there are certain parts of the brain that light up, it does not mean that that's the one area where this is happening. The brain is an interconnected organism, if you will. I mean, it's not an entire thing, but it's its own organ, if you will, that is always interacting at different points. And I know they have there's a newer imaging technique. I'm forgetting the name of it, but fMRI studies, though, people would it's kind of like what we were talking about earlier with the kind of, epigenetics and GWAS, but people just took it and ran with it and just were like, oh my gosh, look, see, this part of the brain lights up. It must be in that part of the brain. Boom, there it is. And now people are like, yeah, it looks pretty. And there's something going on there, but that doesn't mean that's the only thing that's implicated as well. So even in locations in the brain, it doesn't tell you everything that we want to know, unfortunately.
No, absolutely. And again, it boils down to we need reproducibility, and we need an independent way to verify the hypothesis we're testing. Imaging is limited. Right. And we're talking about correlations here, not causations. So we have to curtail the conclusions or make more tentative conclusions about what's going on. The way we can buttress some of these findings is to do psychological testing. So we'll circle back real quick to the amygdala being putatively larger in the average conservative. And again, I'm not saying this is a bad thing. You follow that up with personality profiles or psychological testing, and you, according to some studies, you do find that on average, conservatives have a heightened fear response or a heightened aversion to disturbing imagery. And again, it's not necessarily a bad thing. It's just a very interesting difference in how individuals perceive and react to stimuli.
Yeah, absolutely. So I guess going back to the first part of the question, which what are the aspects? So amygdala is there for fear, but what about some of the other pieces that are there for beliefs and how we understand? There's a lot to uncover there still in the brain, but what can you say, I guess, about beliefs and how we construct them and things like that from various parts of the brain?
Beliefs are very, very complicated. And we know the brain's involved because injury or a disease that affects the brain can alter someone's beliefs. And it can alter behavior in very profound ways as well. There's a very famous study, you might have heard of it, Just to take one of the most bizarre and extreme examples I can think of off the top of my head is that there was a pedophile who, after developing this disturbing behavior, it was found that they had a tumor.
A tumor, yeah, I've heard
growing
that, yeah.
in their brain. Mm-hmm. And when that tumor was removed, remarkably, the pedophilia disappeared. Mm-hmm. Mm-hmm. Tumor grew back, and the pedophilia came back.
Yeah.
So I think this just illustrates, one, how fragile the brain really is, mm-hmm. and mm-hmm. two, how much of it really governs who we are, and three, there's elements of this that we don't control. Mm-hmm. What we consider to be us can change overnight with some kind of brain injury or infection, and we can literally be an entirely different person. Mm-hmm. So I find that a little nightmarish,
yes,
because it raises these interesting philosophical questions as to what do we really mean when we're talking about the self. What does it really represent? Where does it really come from? And it gets into this whole question of, whether people have a soul. If your brain, through a simple little quirk or a tumor, can radically change who you are, what does that mean for the soul, or the supernatural elements that some people claim that we have? Mm-hmm. So that's why I find the brain science and the biology behind the brain so fascinating, mm-hmm. as people have done for hundreds of years. And I think as we continue to explore and investigate the brain, we're going to truly figure out what makes us tick.
Yeah. About the example you provided, I think some people have, from that and maybe other examples, speculate that maybe there's a way of conceptualizing pedophilia as a neurological disorder or disease, as opposed to just this, aspect of personality, or maybe there's a combination. Hard to say. No one really knows. No one says confidently. And then, it does bring questions into focus about free will. And I won't have that conversation now and here, because it's such those conversations become so mired and bogged down in definitions and how do people mean things. But there's definitely, I've said it before on the podcast, I don't really believe in free will necessarily. And I think that there's, when you look at things like this, and you look at how we understand brain behavior relations, how we understand neuroscientific data. I mean, there are things that are happening right before, milliseconds before it actually happens, right? And that we're not controlling that. We're not motivating that. And so I think, and you can make those arguments philosophically. Some people have made those arguments theologically. It's a weird debate, because I think it's getting at the essence of what it means to be human and autonomy and responsibility. But people have had that version of debate pretty much in every field. They have it in psychology, they have it in neuroscience, they have it in theology and philosophy, and et cetera. So it's very interesting how it kind of it's the one thing that kind of bleeds through all of it. I want to ask you two final questions. They're semi-connected, but what do you see as the future of gene research, and any any feelings on CRISPR and the ethical landscape of this? So you can just say what CRISPR is and some of the potential ethical issues that people have raised.
Yeah, sure. And that's why I think, my book is so important. Not to sound like too much of a self-promoter, but I genuinely think this information has got to get out to the general public so we can have informed dialogues and debates about what to do with these technologies that, as you said, fly ahead of us at light speed before our ethics have really thought about them deeply.
Yeah.
Gene editing through tools like CRISPR-Cas9 is one of the prime examples of, we really need to give this some thought yesterday, because the technology's here.
Yeah.
And Just for your listeners' benefit, CRISPR-Cas9 is an enzymatic system that allows scientists to exquisitely edit genes with a level of resolution and accuracy that we've never had before. It's an extraordinarily powerful tool that could potentially hand the reins of evolution to us. And we would be dictating what sort of genes that we want to be passing on into future generations. I don't think that's going to happen anytime soon, personally, but where I do see it coming into play is having a medical benefit. And I think that's how it's going to all start. Going all the way back to sickle cell anemia, there's one single genetic mutation that is responsible for that horrible disease. Theoretically, we have the technology right now to fix that mutation in a very early embryo, utilizing the tools of CRISPR-Cas9. You can just go in there and with like little DNA scissors, clip out the bad part, reinstall a working part, and that individual will be born with all of their other genes intact.
Yeah.
The only exception is they won't have to worry about sickle cell anemia anymore. Mm-hmm. And I think that would be wonderful. You could even argue that it's unethical not to fix it. Mm-hmm. Mm-hmm. So that's how I see the gene editing technology first being implemented. The concern is the slippery slope of where it might go next, because as GWAS studies start to reveal genes and gene networks that might be connected to better IQ scores or better life outcomes, parents are going to want to be interested in tweaking their offspring's genes to maximize their intellectual abilities, for example, maybe their athletic abilities. Mm-hmm. And this gets into the realm of the so-called designer babies.
Mm-hmm. I think so we're still far away from it. I mean, I guess it's, good material for science fiction at the moment, but because there's so many genes in play with those things, we're not there yet. I mean, sickle cell is a pretty specific example. We just, can, replace what's there and then boom, but so many things, even things like eye color, hair, height, all that stuff, is so many genes involved, and we don't know all of the ones that are involved. But I do think that way down the future, if we're able to do this kind of mapping of like, okay, we're pretty sure of all these gene networks, and now we can, once we have that kind of figured out, much as we tried to when we first did the first mapping of the genome, then we could get really into some dark territory there, right? It's kind of designer kinds of things, and so that is a worry. Well, look, Bill, I mean, this has been such a fun conversation. I could keep talking to you for hours and hours and hours. What is the future of this, right? You've talked a lot about good science, communication, which I agree with, but, genes, environment, culture, that's all important. There's so many things going on in the world and with humans. How do we continue to understand these things and then talk about them in a way that is accurate and attainable for people? So I guess what's your kind of final thoughts on where we go and how we should have these conversations about genes and all the rest?
Yeah, I'll share as a final thought some of the revelations that occurred to me during the research and writing of this book, because I had preformed opinions and beliefs about some things that changed as a result of researching this book. Violence being, probably the most clear example. I always thought that, criminal behavior was a premeditated thing. It's totally on the criminal. They're guilty of sin, and it's all on them. But this book, researching it, kind of opened my eyes to engender a great deal more sympathy for what a criminal might be going through. I'm not saying that's a get out of jail free card. We have to protect society, of course. And but what I think the knowledge can be used for is trying to identify biomarkers, if you will, maybe gene predispositions that tell us someone might have behavioral problems, and we need to have an intervention to offset those. Like I said earlier, the so-called warrior gene's only a problem if the child was abused, right? So, obviously the simple common sense scenario is we have to implement social programs that get children out of these dangerous environments so that they don't grow up cursed, if you will, with this burden. And that's a very simple fix. It doesn't involve biology. It doesn't involve gene editing. It involves having a heart. It involves stepping up our social programs to help people who need assistance. So that's one of the major themes I wanted people to take away from this book, was a new appreciation that there's a lot about our personality and behavior that we simply don't control. We didn't get to go shopping for our genes. We didn't get to choose our early environment. We didn't get to go brain shopping. So a lot of this is out of our control. And that, I would hope, would, again, engender some sympathy between people. Because when you interact with someone who is very different than you, you have to understand that if you had their genes and you grew up in their environment, you would be them. And my hope is that readers of this book will gain that appreciation. They will gain that respect of other people. And instead of becoming cold or adverse to those individuals, they will become curious and just learn about what sort of things have happened in their lives to bring them to a certain belief or, a certain political alignment or what have you. And that kind of compassion and respect at the end of the day, I think, is going to help us get along better. We're not going to resolve our differences. I think that's, a hopeless endeavor. People are always going to be different. What we have to try to come to grips with is, appreciating those differences and still maintaining a level of respect amongst one another, despite those disagreements.
Yeah, I agree 110% with everything you just said. And I think that's a really, really good message for people to try and understand. And people get lost in the debate and all of the fighting about certain, is it, the environment or straight genes, and some of that's important. But I think the main elements and the human elements that you're describing there are most, I think, important. The book is called Pleased to Meet Me: Genes, Germs, and the Curious Forces That Make Us Who We Are. Where can people find this, and where can they find you online or your research and all the right places?
Pleased to Meet Me is sold wherever books are sold. So go to your favorite merchant and you can probably find it there. If you want to connect with me, I've written a lot of follow-up articles that build off of some of the content in the book. So if you're interested in that, you can find my writings and other podcasts and appearances at www.authorbillsullivan.com. You can connect with all of my social media channels there as well. You can follow these great conversations I had with Xavier sometimes on Twitter. So you can connect with us both and follow along and join in the discussion.
Yeah, no, it's great. It's been such a wonderful time being able to talk to you in person, and I've really enjoyed the conversation. I think everyone will as well. So just thank you so much for writing such a great book that's very, very attainable for people, and for all of the wonderful thoughts that you've been able to share.
Well, thank you, Xavier. It's been a pleasure coming on your show and having this conversation, and can't thank you enough for providing this forum that allows people who have experience and working knowledge of these biological systems so that we can spread the word.
Yeah, absolutely. Well, thank you so much.